Gene editing for longevity. We've entered a new age of genetic engineering. In the first video, we asked what control really means. Now we'll look at the first part of the control toolbox. This episode traces the milestones in gene editing, from CRISPR's first cuts to today's single-letter rewrites. We'll measure progress the same way engineers do, by tracking accuracy, precision, off-target effects, and the damage caused when errors occur. By following these benchmarks over time, we can start to see the bigger question taking shape. Will gene editing ever become precise and reliable enough to serve as true control? And if so, when? CRISPR-Cas9, the breakthrough of 2012. In 2012, a new tool changed biology forever. CRISPR-Cas9 gave scientists a way to cut DNA exactly where they wanted. For the first time, we could rewrite the code of life with something that looked almost programmable. In just a few years, labs around the world used CRISPR to fix mutations, knock out genes, and even build new crops and cell therapies. It was fast, cheap, and surprisingly easy to use. But the first generation of CRISPR was also rough. It could miss its mark or make double-strand breaks that the cell repaired unpredictably. Typical off-target rates ranged from 1 to 5%, far better than older methods, but too risky for longevity work. Still, compared to earlier gene editing tools, CRISPR slashed the price by orders of magnitude. What once required a $1 million lab could now be done with a graduate student and a laptop. Base editing, a step toward precision. In 2016, scientists introduced base editors, a gene editing tool that can rewrite DNA one letter at a time. So far, these systems can handle 4 of the 12 possible base pair changes. Cytosine editors convert C to T and G to A, while adenine editors swap A to G and T to C. The enzyme complex nicks just one side of the DNA strand. Then a helper enzyme called a deaminase changes the cytosine to uracil. Normally, the cell would fix that uracil back into a C, but the base editor blocks that repair with a chemical tag. When the cell replicates DNA, it reads U as T and inserts an A on the new strand. Subsequent repair leaves a stable Ta pair. This approach works well in dividing cells, but not as well in tissues that rarely divide, like the brain or heart. Base editors still use Cas9 to find their targets, so the number of off-target hits is about the same, but the damage per error is far smaller. Prime editing, expanding the toolkit. In 2019, scientists introduced prime editors, a new generation of tools that could finally write complete DNA changes directly. Instead of cutting or swapping one letter at a time, prime editing works like a true search and replace system for genes. It still uses Cas9 to locate the right spot, but adds a guide carrying both the address and the new sequence to be written. An enzyme called reverse transcriptase then copies that new code straight into the DNA. No double cuts, no waiting for the cell to repair itself. Prime editors can make all 12 possible single base conversions and even insert or delete short sequences with precision. Most importantly, they can finish the edit immediately, even in cells that rarely divide, like those in the brain or heart. That makes them the most flexible and complete gene editing system developed so far. Midstream control, the RNA revolution. In the 2020s, attention turned to a new layer of control, the RNA editing. Instead of rewriting DNA, these tools modify the messenger signals that flow from it. In other words, they let us fine-tune the output without ever touching the source code. This approach includes messenger RNA therapies, RNA interference, and new enzymes that can edit RNA letters directly. Some systems deliver mRNA itself, while others insert short DNA templates that remain outside the genome, yet flood the cell with new RNA messages. These systems can silence a gene, boost it, or even augment a faulty one, all without making permanent changes. Some variants may soon enable true, programmable gene control mechanisms. The COVID-19 mRNA vaccines were the first global proof that this technology works at scale. The next phase, synthetic control circuits. Each new generation of genetic tools has brought a sharper level of control, from cutting DNA, to rewriting it, to regulating its messages in real time. By the 2030s, the next logical step for gene control will be to move beyond single tools. DNA editors, RNA regulators, and protein sensors will begin to share data, linking edits, messages, and responses into coordinated systems. Instead of isolated interventions, we'll see guided networks that respond together, forming the early architecture of synthetic control circuits. Join us in the next video as we move deeper into the science of gene control, because to make all this work, we still need efficient vector systems for delivery, transmembrane proteins for signaling and mapping, and other molecular interfaces that let us communicate with these emerging control systems.